everyone. I welcome you to the 9th session of the seminar series of the Indian Pollinator Initiative, organized by Hema Somanathan from ISA Trivandrum, Professor Belawadi from UAS GKVK in Bangalore, Professor Partip Basu from University of Calcutta, and Joby Joseph from University of Hyderabad, and me, Axel Brockmann from NCBS. I welcome you. So I'm happy and I'm pleased to welcome Xin Zhou from the Chinese Agricultural University in Beijing. He is one of the leader, one has to say, of insect genome sequencing, genome studies, make phylogenetics in insects. He's involved in the big genome projects like 1,000 insect transcriptome evolution that kind of led to a lot of new phylogenies and kind of like phylogenies. And he's involved in the barcoding of Trichoptera. And this leads to his academic career. He did his undergraduates in Beijing University, and then his PhD in Rutgers University, USA, in 2007. And there he was working on Trichoptera, if I'm correct. Then he did a postdoc at the University Institute, Ontario, at the University of Guelph in Canada. That he joined the Beijing Genomic Institute, which is the big institute in kind of sequencing of insect genomes. And I think since a few years he changed to the China Agricultural University. I see it correctly, that is when he really started working on honeybees a lot. And his lab at the Agricultural University, two major projects in honeybees and studying the pollination diversity in the Asian honeybee Apis cerana. And he's whether kind of like different populations of Apis cerana kind of different pollination kind of preferences. And I think this is all with the background of that there is a pollination crisis in China and the interaction of Apis mellifera, which was introduced, and kind of the original honeybee Apis cerana, which is obviously more adapted to pollinating kind of the endemic plants. And this also lead him to I think this is important for us here also, genetic analysis of pollen. And he developed new methods to identify pollen kind of collected from the honeybees. And a project is on the gut microbiota of Apis cerana. Without further ado, I kind of like introduce you to the title of his talk. Today he will talk about gene reuse facilitates rapid radiation and independent adaptation to diverse habitats in the Asian honeybee. Promise you a very interesting talk. Okay, Chin, it's now your part.
Thanks, Axel. I'll start. I'll try to share my screen.
Yeah.
Okay. Can you see my screen?
Yeah.
Okay, good. Well, thanks very much, and I would like to thank the organizers for inviting me. And it's a big pleasure for me to introduce our work on the honeybees. As you might heard from Axel, that I've been mostly working on insects, macroevolution of insects using transcriptomes and genomics. And in fact, this is the first work that we have been doing under species level. So about 4.5 years ago, I started my new position at the China Agricultural University, and it was a time I decided to, start a new model that is more related to agriculture. So I think it was a no-brainer that I moved my research focus on the honeybees, especially the Asian honeybees. So before I start, I would like to start by thanking my co-authors, because obviously this is a collaborative work, and all of the co-authors on this paper have made important contributions. And in addition to that, there are actually a lot of more people who have contributed either knowledge or samples to this work. And I believe that without their help, this paper would not have been possible at all. And of course, at last, I need to thank the funding agencies who give the financial support for us to start this work. My understanding is that I'm the last speaker of this series seminar, and I don't think I need to convince you that the pollinators are important to our agricultural systems. But there are a couple of things I would like to remind you about the honeybees. So basically, we know that the most hot topics about honeybees, on top of their social behaviors, is their functions in pollination and their capability of producing honey. So it is my impression, at least that was my impression in China, that the pollination people and the honey industry people would only rarely talk to each other. But if you think about it, pollen and honey or nectar are actually both major diets for the honeybees. So these things are they're like the two sides of the same coin. So I'll be talking a lot about pollen and honey diets for the Asian honeybees. And I hope that I can show you that the honeybees are capable of adjusting their tendencies to collect these different type of diets as an adaptation to the new environment. Now, from the perspective of agricultural system, the things that made honeybees so important to our agriculture are the facts that all of the apiary species are generalists, meaning that they are capable of eating many different kinds of flowers of different flower species combination. And also, there are a couple of species, namely Apis mellifera and Apis cerana. These two species have really wide distribution range, both across continents. And at the same time, they are capable of capable of becoming adaptive to local habitats. And if you think about it, these two facts are fairly important for not only for the apis species but also for human being, because if the honeybees can only be found in a limited region, for example in the tropical region, and not overlapped with much of the agricultural activities in Asia or other parts of the world, then their contribution to agriculture will be very limited. So our focus has been on Apis cerana for the past 4 years or so. I'm citing a paper from Deborah, published in the year 2000. So basically, based on the mitochondrial data, at least the knowledge back then was that there were 4 lineages of main lineages of Apis cerana. And the whole distribution range of the species cover much of Asia, including Eastern Asia, Southern Asia, and Southeastern Asia. And the climate range covers the tropical, subtropical, and temperate zones. And they can be found in islands, in high mountain valleys, in hills, on plains. And obviously, they can eat a very wide range of diets, many different kinds of flowers as well. And perhaps the most important thing is that the distribution range of the Asian honeybee is largely overlapped with Asia's agricultural systems, which makes them rather influential to more than half of the world populations. And our work is focused on the mainland lineage. So where the the red circle represents the main lineage. Unfortunately, we've seen a significant decline in Apis cerana, with both on the distribution range and population density over the past 100 years. And it is a common belief that this loss of population is primarily due to habitat loss, and perhaps even more seriously by the introduction of Apis mellifera. And also, this is the loss of bee diversity is not unique to China, as we see in the figure on the right. That many other Asian countries are also losing their endemic honeybees. But it's kind of encouraging to see that India gets to keep much of its endemic honeybee diversity, which is a really good thing. Of course, we all know about the colony collapse disorder, the CCD in the America. The U.S. alone is losing 30% of its honeybee colonies per year. And over the past 60 years or so, I think the overall loss is somewhere about 60% of the population. And obviously, there are many causes that could have caused the CCD, among which the lack of nutrition is it sounds like a very bizarre reason. If you think about this. The species survived a very long evolutionary history and expanded from its original distribution range, and managed to conquer many new environments, new habitats, and the species capable of eating many different kinds of flowers. But how could it be, suffering from malnutrition? It seems to be a very conflict results. And so I've been calling this honeybee paradox. So basically, we are interested in understanding how could a historically successful species become challenged at the current time. So to understand that, it would be very interesting to understand how honeybee species, or as colonies, are able to adjust their foraging strategy to deal with diet change. Now, there are many to approach to this question, and the way we took a evolutionary point of view. So we knew that there has been extensive debates on the origin of the genus Apis, but for the common ancestor of all extant Apis species, it is almost the most parsimonious assumption would be that the common ancestor of all extant Apis species would have come from tropical Asia. Now, if that were the case, then we can assume that Apis mellifera and Apis cerana has expanded from their original habitats, and so that they managed to survive in for example, in the much northern region in the temperate zones. And we also know that all honeybee species are generalist, so that they can eat many different kind of flowers, right? And so one of the first questions that we are asking is, what will be the major bottleneck to a generalist bee when they expand their original distribution range? If they can eat many different kind of flowers, what would be the main constraint when they explore the new environment. And the answer to that is not so obvious, but so we had a lot of thinking about it. But when we look at the outskirt of their distribution range, for example, the northern range, the northern limit in northern China and Russia Far East, and if you compare those range with the tropical ones, one of the things that we immediately notice is that these regions, these habitats, have very distinct flower phenology, meaning that in the cold climates, either in the north or in high mountain valleys, you will expect to see flowering, much shortened flowering season. So for example, in the Aba population in China, we only had 3 months of flowers of all years. Okay, and conversely, if you look at the tropical population, then the honeybees can have access to flowers almost all years. There's a huge difference in that in terms of honeybee biology. Because if you think about it, for the honeybees to survive the northern climates, they would have to deal with the harsh winter. And for the northern populations, the outdoor temperature could drop to -30 to -40 in the winter. So we knew that the northern population, the northern colonies, can survive winter by forming a structure called what we call in Chinese a bee ball. So basically, the honeybees would form a ball structure inside their colony, and the core temperature of this bee ball could be as high as 30-some degrees. And obviously, this behavior, this biology would take some serious energy intake, and the energy resource would be obviously would come from honey or honey storage. And obviously, the only way that the colony could store as much honey products is during the flowering season. So if you compare the mountain populations versus the tropical populations, it becomes clear that these populations will have to learn to adjust their energy investment at the colony level in terms of if they want to put in more energy into reproduction or survival of the current populations. And apparently for the mountain or the northern populations, they decided to make the surviving winter as the first priority. And conversely, for the tropical populations, because they have access to the flowers all season, right? And they don't need to store honey for surviving winter. So in that case, they would prefer to collect pollen to increase the reproduction rates. And another interesting fact is that we knew that these collection tendency at the colony level is inheritable. For example, if you put the Hainan, which is the tropical population, if you move the colony to Beijing, which is in the northern part of China, they will fail to survive, and they just simply refuse to collect or restore or store honey for winter. And so these are the known facts, but we don't really know what the molecular mechanism to the underlining all these interesting biology. And another thing that is challenging to understand is that, so we knew that there must be a molecular mechanism that determine this behavior, but how could natural selection work upon the colony level when the molecules only determine individual behaviors, right? So there are a couple more things that we knew about. First of all, as a eusocial insects, we knew that honeybees are capable of, they actually have very sophisticated labor division system, including foraging labors. So we know that some collectors would prefer or have stronger tendency to collect a specific type of food. For example, some would collect water, while others would prefer to collect pollen or nectar. And we learned from a paper that was published in the year 2000 that the foraging division of foragers when they were at 2 to 3 weeks old, it was predetermined by the level of sucrose sensitivity when they were 1 week old, when they were still a nurse bee. So, which means that, for example, if a worker had a lower sucrose sensitivity, when they grow up, when they started to collect, they would tend to collect nectar instead of pollen. Of course, we know that they could switch based on the environmental conditions or internal needs of the colony, but the tendency were there. So we know that if the worker has a different level of sucrose sensitivity, they tend to collect different things. And another fact that we knew of was because honeybees are an example of extreme polyandry, so workers, including foragers, would have different fathers. And we also knew that foragers from different patron lines would have different tendency to collect various foods. For example, all the daughter from one father would tend to collect more pollen, and while all the daughters from another patron lines would have a stronger tendency to collect nectar. So this is a very neat system because this means that if we knew the molecular mechanism that determines the sucrose sensitivity, and natural system will then have a way to work on the colony level, because the patron lines that have the perfect foraging strategy would be strongly selected in a given condition. So given all that we know, here are our two hypotheses before we start our population study. So because we know that the Asian honeybees have a really wide range and they can deal with all sorts of different kinds of habitats, and we assume that the current populations must have undergone parallel adaptation during a range change. And secondly, we would assume that the genes involved in foraging division must be repeatedly selected, what we call gene reuse. So in this way, if we find a particular set of genes that are constantly or highly repeatedly selected among these independently derived populations or parallel adaptation, then there's a high chance that we can actually find the genes that are responsible for foraging division. I think I can stop here if there's any questions. I know I've spent quite a while to explain the background, but I think this is the very important information to understand our experimental design and the results.
Are there any questions? There's no question. I think you were very clear. So stopping.
Okay. I'll
continue.
Sorry?
I have questions, but that's more for the end discussion.
Okay. So here's what we did. Basically, we sequenced we carry out whole genome sequencing for more than 300 individuals of Apis cerana in and around China. So we had one individual worker representing a colony from a large range of this species. And from this data set, we got 4.5 million SNPs. So these are single nucleotide variations across the genome sequence. And so I don't want to get into all the details of the analytical pipelines. So I'm going to just skip to the results of our findings. But I don't think we did any particularly fancy stuff. So most of the analysis that we did with this paper was more or less a standard population genomics analysis. But from these analysis, the first thing that we noticed is that among the large range of the species distribution of all the populations that we sequenced, there were 7 genetically unique population groups. And as you can see here in the figure, so basically there are this central group and all the colorful dots, which represents what we call peripheral groups. The central group covers much of the distribution range of the mainland species, of the mainland lineage. So all the populations, all the individuals that we sequenced represent a single genetically unique population group. And we call it single because we didn't notice any apparent internal structure within this large population. And conversely, if you look at the peripheral ones, the colorful ones, these ones has formed a mutually monophyletic group cluster on the neighbor joining tree. And another thing that we noticed that all of these peripheral groups, all of these colorful ones, have independently derived from the central group. And also, we summarized the genetic diversity. We calculated the genetic diversities for these 7 groups. And the first thing that we noticed is that the genetic diversity, which we calculated in 3 ways, for the central group is always significantly higher than any of the peripheral groups. And when we started comparing the genetic divergence between these population groups, we noticed that first of all, the central group, the divergence between the central groups and the peripheral group is always smaller than those between the peripheral groups, which means the peripheral groups, the central group is always the genetically more similar group to the peripheral groups. And this divergence is also, it has nothing to do with the population size, because if we divide the central groups into provinces or local populations, you only see even smaller divergence within the group. And also we have evidence to show that each of the peripheral group has ongoing genetic flow with the central group. And another thing I would like to mention is that the average genetic divergence between the Chinese Apis cerana groups, between the central group and the peripheral groups, is actually equivalent to those between the Apis mellifera subspecies. Actually, it's even larger than subspecies divergence. So technically, I think what we're seeing here are subspecies level divergence, local population groups. But apparently defining subspecies is not a priority for this work. So we've been calling them population groups in our study. And also this population structure is also confirmed by evidence from mitogenomes. So these are mitochondrial, the full length of mitochondrial CO1 gene, and also by morphology of the four wings. We did a classic morphomatrix analysis with the four wing characters. So all these clustering pattern would agree with the what we found with the genomics data. And lastly, we want to understand how are these population groups related to each other. So we built a maximum likelihood tree in addition to the neighbor domain tree. And again, the results strongly suggest that each of the peripheral group actually derived from the central group, which is represented by the black line here. And all these peripheral groups derived independently. So there's no direct relationship between any of the peripheral groups. So from these part of the results that that I have shown so far, our conclusion was that first of all, we have 7 subspecies level population groups within China, within the range of our study. And each of the peripheral groups independently derived from the central groups, which is the common ancestor of these peripheral populations. And so we went further to understand how the population have evolved, in particular how they respond to climate change. So we calculated the effective population sizes for all these population groups, and the results was quite interesting, because if you think of how the population sizes change over time, it was almost perfectly mapping with the temperature curve. So at about 100,000 years ago, which is at the end of the which is I think this is the beginning of the last glacial period. So the temperature keeps dropping for almost 80,000 years, and all of the population sizes kept decreasing until to the point where the maximum the maximum of the last glacial period, when the temperature started to climb, and the central population, the population size of the central population started to follow. Conversely, all the other populations kept decreasing until very recently they started to expand. Okay, so we also estimated the historical distribution range for the present time and for the LGM. It appears that at the maximum of the LGM, the population distribution range for the mainland, well, actually, I think this might have been all Apis cerana. So much of its population has retracted to what is currently recognized as the Sunderland, and including some regions that were exposed in the geological time. So our interpretation from these results was that the mainland Asian honeybee has undergone what we call centrifugal speciation model. So basically, if you think about the central population as an ancestor, this ancestor group has been pumping out peripheral populations, and which have been left out in the peripheral region. And later on, the central populations start to contract and re-expand. And the current population structure and pattern that we see today is the result of a secondary contact. Okay. And if we map all the current peripheral populations in a geographic map, it becomes more clear that all of the, well, this is just part of the peripheral populations, but all the populations that we found on the edge of the Qinghai-Tibet plateau, it seems that they all have been trapped inside the valley. They haven't really come to the plateau, actually, but they were all living inside deep inside the valley. So it appears that these valleys have been serving as refugia in the cold climate. Now, now that we have the population structure redefined, and we started to look at some of the biology or biological traits of these local populations, and we noticed that as expected, the tropical population group in Hainan group has a very strong tendency to collect pollen instead of nectar. And all the northern population and the valley populations have much stronger tendency to collect nectar. So these have been a lot of these have been recognized as good strains of honeybee strains in China, which could produce good quality of honey. Now, so based on these results, it appears that our first assumption, our first hypothesis, is supported by the evidence that we collected. So basically, the current population is a result. The current population pattern and structure is a result of parallel adaptation during the range expansion of a common ancestor. I think this is a good time to make a break too. If there's any question, I'll be happy to answer. So I should click on this, I assume.
I think Axel has temporarily lost connection, but
Okay, I can see two.
Yes, Professor Bellavadi, can you ask your question?
I have one question. That is, you mean to say that there is speciation that is happening in the borders of the distribution?
Well, this is
whether Apis cerana is, yeah, the populations are ultimately going to become separate species.
Well, I'm actually not suggesting speciation ongoing. Yeah. I'm saying that the populations are apparently undergoing a speciation process with the yeah.
Now, in the long run, you expect them to become, unless there is going to be again mixing of the populations.
Yeah, there could be many different future for this peripheral populations. As you suggested, that they could be intermingled again with their ancestors, with the ancestors that have formed a secondary contact. In fact, we have detected ongoing gene flows between these populations, which is also common. I mean, these are in fact the same species, right? But also there could be possibility that if the peripheral populations are being kept for longer run in this local region, and the reproduction isolation could be further strengthened, and then new species might be possible.
Because I think in Deborah Smith's talk, she was mentioning that there are far more number of species of Apis cerana than what we think of the Asian species. So yeah, that's one. Okay, thank you.
Thank you.
Yeah, can I ask a question? Yeah, I think it's admitted. Yeah, just regarding the nectar collecting bees in the high altitudes, isn't that due to also because the nectar is more viscous in high altitude? So isn't that a way of adapting? You know, they need more nectar, but the nectar is more viscous, not fluidy. Is that a reason why they are going for the lineages in the high altitude are more nectar collecting than the pollen collecting ones?
Well, I think these reasons are not mutually exclusive. So our interest has been on how honeybees would adjust their strategies into collecting different things. But it's also possible that, as you suggested, that the flower features could be different, and so that the quality or quantity of the honey that they collected are better than other populations from other regions. But I don't think I've seen any detailed studies on this front in China, at least.
Okay, thank you.
Oh, I can see my microphone is unmuted. Can you hear me?
Yes.
I was just wondering, where do you think these subspecies or these different subpopulations will be going in the future with current climate change predictions? So do you think some of the groups will be expanding or range contracting? I'll just have to say that I'm currently modeling Asian honeybee distributions in future climate change model scenarios, but I haven't taken into account subspecies like you've been doing with your modeling.
Yeah, I think that is a very interesting question. That is very actually the kind of question we would be interested in answering in the future. I don't think there's a simple answer to that, unfortunately. Apparently, the most serious challenge in China would be the introduction of the Western honeybees, and so I think to this point, for most of the peripheral populations that we managed to identify in our study, the Western honeybee is not yet a major challenge or in their local habitats yet. But that being said, we've seen that in many places that the bee farmers started to replace their endemic bees with the Western honeybee, simply because they're easier to manage. So I think to the Chinese populations, I think that the more immediate challenge would actually come from the introduction of Western honeybees.
Okay.
Yeah. But I agree that it is a very interesting question to answer in terms of the future for this adapted local populations.
Great, thank you.
Question. Can I ask a question?
Sure, please.
I'm Hema. Do you find that any of your populations, either the central population or the peripheral populations, do you find any differences in, say, colony size or in the ontogeny of tasks?
I think the short answer is I don't know. As I explained earlier in the start of my talk, we've only started working on Asian honeybees for 4 years, and we were lucky to have all these samples from our collaborators. So we only started to study their biology and ecology only very recently. But there are some extensive study in China in the past on some of the local populations. So we know that, for example, the slides that I gave in terms of tendency to pollen versus honey collection, and those were from historical research. And so we knew some of the biology there, but not much, I have to say. So I guess I have to say I don't know at this point.
Yeah. Thank you. Yeah.
Oh, hi.
Axel is offline, so I will moderate until he's back. Please go ahead. Yeah.
Okay. This is more of a comment than a question. I just want to say that, for many years, we've been fascinated by the effects of climate changes and the glaciation cycles and sea level changes in Sundaland area, where you obviously have land masses joining and separating. But it's really fascinating to see what's happening where it's less obvious on the Asian mainland, what changes are, what the effects of climatic history is on these populations, and that's really a lovely new aspect of studying the Asian cavity nesting bees.
Well, thanks for the comments, Deborah. Okay, so are there still questions? If not,
yeah, I think you can continue, and then we can take questions at the end.
Yeah. Okay.
Thank you. Yeah.
All right. So I'm going to continue with the rest of our results. So now that we have a better understanding of the population structure and history, then we started to try to understand whether there are any genes that were under repeated selection as we expected. So we did a whole genome screening for all these individuals that represents all these population groups. The first thing that we noticed is that for the majority of the genes under selection, they can be only found in a single population group, which is not surprising because these were in fact independent expansion events. So each of these rows represent a peripheral population, right? So the dots, the two dots that are connecting the populations means that there were a particular number of genes that are being selected in these two independently adapted populations, right? So I was saying that most of the genes that were under a selection were only found in a single population group. So for us, the most interesting part is the ones that are being selected in independent population groups, right? As it turned out, there were a significant proportion of genes under selection in more than one independent group, and this number is significantly higher than what can be expected by random. And among all these genes, all these selected genes, a lot of them we can infer that a lot of the selected genes have something to do with their adaptation to habitats. For example, they have something to do with their appetite, with their circadian rhythm, or olfactory receptions, and so on and so forth. And perhaps the most interesting thing for us, we've been looking for the gene that had been almost constantly selected during the range expansion of the honeybees, right? And so there was actually one gene that was almost always being selected in all these populations. In fact, the only exception here in the Taiwan population, it was actually detected in one of the methods that we used. So it turned out that this gene was a perfect exemplar that we can examine for his function. Now, we didn't really know what the gene was until we construct a gene phylogeny using all the sequences that are similar to the one that we had and downloaded from GenBank. And we also looked at some of the sequence traits or characteristics of this particular gene sequence. And from all this evidence that we conclude that this particular gene, the gene that was always being selected during expansion, it was actually turned out to be a star gene, because it has been studied in many kind of model systems, like a human, like mouse, like Drosophila, the mosquito, and the C. elegans. It turned out to be a weight loss related gene. Because it has a lot to do with the promoting the feeding motivation, or the increasing tolerance to bitter foods, or the mitigating anxiety effects, or fear, or stress. In short, it almost certain that this gene, the homologous genes, have something to do with eating, or diet, or foraging. So it was possible to us to assume that this gene were actually doing something during the range expansion of the Asian honeybees. So were they assisting adaptation to the new diets, or they assisting the foraging division? So we went further to test to do some functional test. So the first thing that we looked at was the expression level of the LKR gene in the different tissues of the honeybee worker. So basically, if you look at this figure, the LKR expression level was significantly higher in both antennae and brain when compared to the thorax and gut. So we knew that these are the primary foraging related organs for the insects, right? And another thing that we noticed is that the level of LKR expression was also significantly higher in both antennae and brain of the forager when compared to the nurse bees. So again, these results seems to be compatible to our assumption that this gene is doing something for the foraging behavior in the honeybees. And then we did a RNA interference experiment on the LKR gene. So the RNAi experiment has significantly decreased the LKR expression in the brain of both forager and foreigners in the brain. So the effect could be seen after the application of RNAi in 72 hours. And so the knocking down of LKR expression has a direct effects on their behavior. So we did a classic PER test on the knocked down the workers. So when you knock down the RNAi level, their sucrose sensitivity level has also significantly decreased, which is in accordance to what we have expected with the function of the LKR gene. So remind you that we already knew that the foraging division of these foragers was determined by the level of sucrose sensitivity when they were younger, right? So which means if the LKR expression level can determine the sucrose sensitivity, these systems will eventually decide, what kind of foods the foragers will turn out to collect. And so we did another additional test with the pollen and nectar foragers. So we collect these different foragers on their foods, and we test and we measure the expression level of LKR in their brain. And again, it appears that the expression level in their brain is much more lower in the nectar collectors, which is what we expected with our previous assumption. So with these results, we came to the conclusion that there are actually multiple genes being repeatedly selected during the parallel adaptation of the Asian honeybee populations. Of these genes, the LKR is involved in foraging division. So it seems that both of our hypotheses have been supported with our evidence, but when we think about the whole picture, it's almost like we have opened a can of worm, where the whole system is rather complicated and the whole logic chain is rather long. So what we managed to do so far is only some part of the whole picture, and it will apparently take a lot of more efforts to actually prove that this whole system actually work as what we thought. But there are a few things that I think we've learned from our study, especially how the honeybee species have survived diverse habitats with just a single set of genome. Okay, it seems like that the gene reuse is a common mechanism in independent population expansion and adaptation of the species. And the LKR gene likely plays an important role in foraging task division, a molecular mechanism enabling natural selection to work upon colony level, and which is an adaptation to the changing flower phenology. And we've actually did some analysis on the selection types of the natural selection that we found in on the genome, and also we've done some preliminary analysis on the possible regulatory ways that are related to the LKR regulation. But for the sake of time, I didn't really include those results in the talk. But all of these results are included in the paper, which is provisionally accepted. So hopefully it will go through the publication procedure very soon. So you'll be able to see those part of the results. And the last piece is there are a few more things that we learned from the Asian honeybee model. First of all, I think when we talk about taxonomy, especially the definition for honeybee subspecies or species, I think it's obvious that we need to take evolution into consideration. So those that deserve that have undergone evolutionary process as a unit would deserve some sort of a taxonomic assignments, either species or subspecies. And also, it appears that the centrifugal speciation seems to be an important model for the Asian honeybees, especially for the mainland Asian honeybees. We don't have data for the Sunderland, for the more southern populations, but it seems to me it might be also the case for other population as well. And also, as I said, gene reuse is probably a common mechanism when a species of wide range become adapted to new environment. And also, the natural selection would work through use sociality, so that the selection working on individual level will become will eventually come as a beneficial output for the colony. And for if we consider pollination and honey production, these are actually both adaptive traits that are developed during the honeybee evolution. So there might be a way to, as long as human beings understand how honeybees manage to balance these needs, there might be a way to either promote pollination service or increase their honeybee production. Perhaps in the future, I don't know. And the last piece of my my slides is that I'm really interested in understanding the evolutionary mechanism and history of Apis cerana, but we're facing a big challenge here, not just in the science itself, but also we have basically we need to find a better way to present or find a better angle to study the Asian honeybee, because the most of the knowledge that we learn from for the honeybee were based on the Western species. And at least to me, the Apis cerana is an excellent model for studying natural history, for studying natural evolution and adaptation. So hopefully there could be more people start working on this exciting new model. And here's my team. We have many prospective and talent young people. And lastly, I would like to thank you for your attention. And I would like to take questions. Thank you.
Are there any questions? I see a raised hand from NS Bhatt. You can ask your question.
I can't hear anything. Hello?
Tommy? I think there's a raised hand. NS Bhatt, to ask your question. Hello.
I think Joby is trying to ask a question, but I can't hear you. Is this just me
You
or?
need to unmute yourself.
Yeah,
Mute your mic.
Can you hear me? Can you hear me?
Yeah.
Oh, here we go. Yeah.
Yeah. So I have not read the paper which talked about the switching the roles between nectar, pollen, and water. So how easy is it for the switch? Because we see it quite often happening in our mellifera colony. So is it very difficult to switch in cerana?
I don't know how to answer this because I don't know how to measure the level of difficulty. But
Yeah, so
why
the way we see it is, suppose we mark them with the sucrose feeder, then we can see the same bees appearing in the pollen feeder and at the water collecting point. That's the way. That's the only evidence I have. So.
So I think the study back in the year 2000 was the way how they did it was they actually marked every individual that they tested for psychosensitivity. They observe what they would collect. That's how they categorize those the foraging tasks.
Yeah.
But I think, as I said, the level of sacral sensitivity only determines the tendency. For yeah. So there will be individuals that who wouldn't follow this rule.
Okay.
And there will be cases where some of the predetermined foragers would start to collect different foods at some point. The mechanisms are still unknown, but we know that these, from an evolutionary point of view, that these flexibility will actually be beneficial to the colony. Yeah, but obviously this is a very complicated system. This is what we're trying to understand.
Yeah. Thank you.
Are there any more questions?
So before I ask my question, a quick point, which is that life size seems to have got updated and including a chat box now. So like people who are having connection difficulty can actually enter it in the chat box. Somehow it doesn't seem to have notifications. So it has to be open to see what people are. But yeah, those with connection trouble could, I guess, use the chat box. And also the continued screen sharing is causing echoes. So it'll be a good idea to like turn off the screen share.
For me or for
your screen sharing. So anyway, I'll ask my question after Professor NS Bhatt has typed his question in the chat box. So it'll be visible in the bottom menu. So I'll just wait for that question to be asked and I'll go last.
I think, while he types his question, we can take other questions.
Okay, I can ask my question.
Go ahead.
Basically, like rather than give a so this LKR expression, could it be related to the distance calibration function, which is basically the waggle duration to food source distance calibration curve? So I mean, generally it's seen that there's a less steep distance calibration curve. I guess the experiments that study this are mainly look at the nectar foraging. So so considering, like, for example, in mellifera Fera and cerana. In both, the colder climate subspecies appear to have less steep and distance calibration curves that have a greater foraging range. I mean, there's not too many data points for these observations, but there's a small amount of data points, and also the LKR expression level determines the tendency to forage for nectar as opposed to pollen. And it is also being discussed in today's talk that colder climate adapted populations tend to forage more for nectar and warmer climate more for pollen. So this is why I'm wondering if LKR expression could be one of the factors underlying the population-specific distance calibration curves, if they exist.
I'm not quite sure if I understand the full question, but I think you're suggesting that the LKR might also be responsible for the foraging distance related calibration. Right?
Yeah, I'm saying that because it's like associated with, because since specific LKR expression levels might be associated with different foraging behavior based on the climate, based on the discussion. So this was my question, is basically LKR and the distance calibration curve, could there be a relationship? And everything else I said was basically the background for my question. So that's basically, yeah.
Sorry, my internet connection is broken. But let me add something to that, because I think what I'm trying to say is that what we found was LKR is apparently related to sucrose sensitivity in the bee brain, but that doesn't mean that it is the only gene or it is the only pathways that are determining or that are related to the foraging behavior. In fact, we actually assume that there are many different ways that could change the LKR expression level, so that the bees would be altered in terms of foraging strategy. And now what there might be all sorts of different factors that could have impacts on this. It's just simply that we don't know that much about the system. But it will be interesting to test, some of the individual factors.
Right. Okay.
Any other questions? Pinu, do you want to ask a question?
Yeah, Hema.
Please go ahead.
Yeah, thank you, Hema. The question is, when this LKR gene expressing, have you noticed any other set of genes upregulating or downregulating correlatively, which are really involved in the foraging? Have you noticed any other set of genes which are really involved in foraging, upregulating or downregulating when this is expressed?
Right, that is an excellent question. We have actually started to look at the transcriptome, the different expression level of transcriptomes, but that analysis is not included in the current paper, which is why I did not include it in the presentation. But that being said, as I said before, we actually expected that there are multiple factors that could change the expression level of LKR. So it would actually be a very difficult task to control all these variables at a time, so that we can only we could manage to test a direct effect of one particular factor to the expression level of LKR. Yeah, we put a lot of thoughts in this, including, there are a lot of things that I didn't manage to talk about. For example, the genetic background with differences within the colony, which will actually have a major influence on the how LKR will interact with the foraging division of a particular forager. But my short answer to your question is that no, we haven't. I don't have the results at hand, but yeah, that's something that we're looking at.
Can I also just let you know my observation? Although Apis cerana at the species level is polylactic, that means the members or the workers are foraging many species. At the individual level, they are not. Once the resources are getting over, they decide to move to the next species, and maybe they may be going for the nectar foraging from the pollen foraging. So do you think in that way this LKR expression is changing in such bees where they are switching from pollen to nectar foraging, or vice versa?
This is, again, another excellent question. We've actually started to look at some of the data for real allele frequencies in some of the peripheral populations, and the results turned out to be very complicated. We had assumed that when you sequence the LKR expression, then you would see a immediate division in between their foragers and between their pollen foragers and their nectar foragers. But it turned out to be more complicated than that. We actually assumed that for some of the peripheral populations, the allele, LKR allele, have been selected to a single allele type, so that this allele type can actually produce labor divisions, produce foraging divisions, aiming for different foods. So apparently, LKR, although it is playing a central role in this determining system, it's not. Again, it is not the only gene that are responsible for this. So, giving everything else equal, there should be some other genes that involved to determine, which specific individual would actually collect either pollen or honey. I'm not sure if I answered your question correctly.
Yeah, you're right. My final question, of course, you have shown that bar chart with the different genes and their expression level, etc. Why did you choose LKR? Having seen its function in other organisms, slightly related or it is unrelated, why did you pick up LKR gene specifically in this study? Yeah, I can see this LKR gene expression over here in the bar plot on the right-hand side. Compared to so many other genes, expression is pretty low. And why did you choose this LKR gene? Any specific reason for that?
If we're looking at the same slide, this figure actually doesn't show the expression level. This is the genes that are under selection in different peripheral populations. So if you see a dot here in this, it says that this gene has been selected in this particular peripheral population group. So these results actually suggest that this LKR gene has been selected among all these populations, right? So this will be something that we are interested in look at, because, right?
Yeah. Thank you so much. Yeah.
And also, because this gene has been studied in other systems, and seems to be related to foraging. So.
Okay, great. Thank you.
I think there are no more raised hands, but there are some questions in the chat box. Would you like to look at the chat box? You see those 3 dots on the right-hand side. If you click on that, see the chat box, and there are a few queries there.
Sorry, I can't. Which chat box?
You see on the right-hand side of your screen, the bottom right, there's a circle on the extreme right has 3 points, 3 dots. If you click on that,
okay.
Open it, you will see
Chat.
something called meeting chat, and there are a lot of questions. You can do it. I can also read out the questions to you.
Sorry,
Can I read out the questions to you?
Yes, please.
So, one question from Pardeep Kumar, which says that managed pollination practices are commonly used to improve pollination efficiency, but I don't understand what the question is there. So maybe that was just a comment.
It's a comment.
Okay, so there's another question. There's a question from NS Bhatt. Yes, we've observed higher flower constancy in wild bee species compared to hive bees. What is your opinion with respect to gene regulation?
Sorry, could you please repeat that question?
Yeah, so he says that he has observed higher flower constancy in wild bee species compared to hive bees. I think he means the ones kept in boxes. So what is your opinion with respect to gene regulation?
Well, that is a fairly big question. My impression was that the Asian honeybee has been less domesticated when compared to the Western honeybees. So basically, what we have in China is the capped or the managed bees, the Asian honeybees. They have a tendency to escape. So whenever they feel threat or they feel that the food supply is not sufficient for survival, they will tend to escape. So that means there has been almost constant gene exchange with the wild populations. In fact, in our work, one of our biggest challenge was to actually identify which populations are the real wild population. Because managed bee farms are actually quite popular in China for the domestic bees. So I don't think there is an easy way to tell the differences in terms of gene expression between the wild and domestic populations, the colonies. Have I answered the question?
I can't hear anything.
Hello? Can you hear me? Hello? Hello, can you hear me?
Hello. Hi, I think even Hema might be having connectivity issue, but I can see her online still. So at the bottom that panel, you can see where your mouse is hovering right now. Can you just click on that? I think you should be able to see the chat window. So there is that, no, the third, yeah, the one next to that, yeah, that one, yes.
This one?
Okay, maybe.
I can't see any.
Yes. Yeah. Maybe I can read out some of the other questions.
Yes, please.
So Jose Matthew has asked a general question. So his question is as follows. You showed maps of honeybee decline in India, China, and other countries. Both in India and China, decline is more in the northern half. Is this a climate, that is tropic versus temperate issue, or genetic differences between subpopulations, or differences in food availability, or human impact? Or is this just an artifact? What do you think? What is the most important factor in colony collapse? That's his question.
Well, that is an excellent observation. I actually didn't notice this, and I don't think I'm the right person to answer this question. I have not had the opportunity to actually study populations of the Indian populations, especially from the south. So I don't have any genetic evidence for that. Maybe Deborah has some data from that region. I don't know. So I can't. I don't know the answer to that question.
Okay. So I think Professor Bhatt's question was, NS Bhatt's question was as follows. Diversity in cerana is blessing in disguise. Perhaps such diversity could have saved mellifera from colony collapse disorder. I think he's asking for your opinion on that.
Sorry, the connection is broken. Could you repeat?
The question was if the diversity which is present in Apis cerana, if there was corresponding diversity in Apis mellifera, if they would have been saved from colony collapse disorder.
So the question was if sorry, I can't understand the question. If the population of the Apis cerana has been saved?
No. So the question was, so if Apis mellifera had the kind of diversity that you have shown to exist in Apis cerana, if they would have been saved from colony collapse disorder.
Well, it seems like a very complicated question. It has a lot to do with the causes of CCD, and I think genetic diversity is only one of them, if there's any evidence to show at all. But I do not agree that Apis mellifera has lower diversity compared to Apis cerana, because there are actually dozens of subspecies identified for the Western honeybee, and many of those were found only in Africa, and also a lot of subspecies found in Europe. The colonies that are used in North America are all introduced in about 100 years. So there might be a chance that the diversity of those populations, those specific populations, are decreasing, which could be a factor. But I don't think I'm the right person to answer the questions on Apis mellifera.
Okay. Moving on to the next question. 1 second. So if the level of, so this is a question from, I'm sorry, I may be pronouncing the name wrong, from Gebretinse Halu. So the question is as follows. If the level of sucrose sensitivity and the tendency of nectar pollen collection are inheritable as mentioned in the background, how is the foraging behavior associated with the genetic backgrounds?
Oh, that's a good question. A simple answer is we don't know. We don't have data to support that. The observation of collecting tendency and inheritability were based on classic research methods, and there was no genomic data or genetic data to accompany that. So we actually had a plan that we would collect both genetic data and behavioral traits data from the same colony and compare that and do some sort of a association work with those data. I think to answer that question, you need basically you need some specific designed experiment that can connect biological traits with the genomics.
Okay, Professor N.S. Bhatt had asked another question. His question is the following. Geometric framework is known in larvae of certain insects for nutrient regulation. Do you think bees chose the nutrition level of the forage during foraging?
I would like to think that I do, but I don't have evidence. In fact, we have a way to quantify pollen contents from the bee brats, and we have planned. We are currently working on that project actually to see is there any correlation between the kind of pollen or the proportion of pollen that the bees will collect when compared to their reproduction cycle. So the assumption was that when the colonies start to produce a large amount of larvae, the colony would actually tend to collect the pollen species that with enriched protein contents. So because now we are able to tell what pollen species are in the bee bread, we then could make all these connections. But the samples are being collected as we speak, but we don't have the data. And hopefully I have something to show in the future.
I think.
Okay, so I think I have more or less exhausted questions in the chat box. There is a comment on about stereotype behavior. Pollen collection is always proportional to brood to support above. Stereotype behavior in honeybees has its basis for genetically controlled behavior. I'm not exactly sure what the question is over here. So I think there are no more new questions in the chat box. And let me check if there are any more new raised hands. There is one raised hand from Vidur.
Yeah,
Vidur?
So I would like to try to ask my question more clearly than the last time. So the basic question is whether perhaps not LKR specifically, but genes like LKR that are involved in foraging behavior in general, and like aspects like whether the bee looks for nectar or for pollen. That's the basic whether such genes whether the same general set of genes is also the basis for the distance calibration function. And the reason for asking this is that, like earlier in the talk, it was discussed that that cold climate adapted bees tend to collect nectar more, and hot climate populations collect pollen more, and this is determined by LKR expression. And now it's also known generally that there's not too many data points for this, but it's known from some studies that cold adapted populations in the same species tend to have less steep distance calibration curves and hence greater foraging ranges than in tropical populations. So that's why I'm just like it's not, so I understand that like LKR wouldn't be the only player, but a gene, but like LKR and genes that behave that operates similarly, can we like say something about their potential involvement in the distance calibration function, that is the waggle phase duration versus distance to the so that's my attempt to more clearly say the different backgrounds I'm stitching together to ask my question. So I hope that's a bit clearer now.
Yeah, thank you. I think I understand better. I don't think I have much to add to my previous answer. Obviously, I'm not familiar with the distance calibration system, but as I said earlier, I think LKR is involved in what the key notes in regulating sexual sensitivity, which in turn determines the labor division, foraging labor division. But then what could have impacts on the LKR expression remains unknown. So the what regulates their expression level is still needs to be studied. So I tend to think that there could be many factors, external stimuli that could change the expression level. But what exactly, I don't know. It could be the distance-based calibration system. But how would it work? I don't know either. But
okay. So based on your answer, I suppose one can infer that that it could be that there's something upstream in the cascade of which LKR expression is a part, and that upstream thing could have a different branch that goes into something like the distance calibration function. The upstream thing could be to do with foraging in general, and it could like affect different things through different branches, and the LKR expression might be in one of the branches. Is that something I can infer from your answer?
Yeah, I think that could be very possible.
Okay. Thank you.
Okay, so I think a lot of questions, but for the first time today unfortunately in your talk, we had a lot of with connectivity at various levels, I think. But despite that, I think we all got the message and we understand that exciting. We have Apis cerana here in our backyards, which are less explored. So I hope that, this talk is going to stimulate people to go out and look more at Apis cerana in India. So thank you so much for this really fascinating lecture. I foresee that I'm going to be in touch with you. I have some more questions, but I will email you about it. Thank you so much for spending your time talking to us, for sharing your very recent work.
Yeah.
And we look forward to reading the paper.
Yeah, thank you. Thank you all for listening.
Thank you. Thank you so much.
Thank you.
Yeah. Bye. Bye.
Bye-bye.
Thank you. The meeting